So in this video, we'll be finishing off looking at how materials get in and out of cells. Here we've got two solutions. One is hypertonic. There's a whole bunch of sodium bicarbonate or baking soda dissolved in this water. This is just tap water. I know it looks cloudy. It's just because it's been sitting all night. This piece of celery, though, is nice and crisp. Wouldn't eat it, frankly, but it is nice and crisp. This piece in the hypertonic solution is shriveled. So indeed, from the central vacuole, water has been drawn out, basically sucked out of the cells. And that is, movement of water is called osmosis. So in either case, water's been sucked out and water's been sucked in. In either case, it's a movement by diffusion of water, which is called osmosis. So we'll be looking at diffusion, just simple diffusion. Facilitated diffusion doesn't require energy, either of those two. As part of the diffusion, we'll be talking about osmosis, which we're just showing you right here, and active transport. Now, active transport, for example, your neurons right now, to fire, to think, to move, your neurons need to basically be pumping sodium and potassium, basically potassium in and sodium out. Now, that pumping action needs energy. So ultimately, even just doing something like this is cells needing energy, and of course active transport. Also with these plants right here, they're using active transport to suck nutrients into the roots, which that actually helps them suck water up into the plant. Okay, so let's get going.
All right, so let's get underway. We're going to be looking at how do particles move in and out across the semi-permeable membrane, the cell. There's two that require no energy, two types, diffusion and facilitated diffusion. Within diffusion, the movement of water, osmosis, is a type of diffusion. Facilitated diffusion, like you can see here, you need some kind of a channel. The other major type is active transport, which does indeed require energy. Yeah, so let's pull up our notes here. Oh, just really quickly, look beside me here. We've got how diffusion works. You go from high concentration, so we got some burnt toast. Eventually, the whole house smells like burnt toast. Okay, let me share my screen here so we can see also where we're at. So in the course, we're right here, day 9. This includes an osmosis lab. Once you complete this, you'll not only be able to do the lab, but also that cell membrane and transport assignment. We'll have a review video for you to watch, and then you'll be able to complete the next quiz, the final assignment, and then do the exam for this unit that we're doing in biology on cells. But yeah, let's begin. This is our focus today. Transport across the cell membrane. We want to know these three processes, looking at osmosis and something called endo and exocytosis. Some things are just too large to get into the cell. So the cell membrane itself, not pictured here, we'll be looking at this. The cell membrane itself can open up and either let things out or let things, oops, got that switched. This is to let things in, and this is out, exo, right? Exit. But yeah, we'll be looking at that. Here's a good visual of those three key processes: simple diffusion, facilitated diffusion, and active transport. Let's take a really quick look at a video of diffusion. Particles are always in motion. That is the main reason why diffusion works, and cells take advantage of that. And some things can just easily diffuse across the cell membrane. Oxygen can easily go in. Carbon dioxide can easily go out. Water takes a little bit, but water can also move across the cell membrane, and we're going to definitely be looking at that today. Okay, so let's take a look more carefully at particle motion across a cell membrane. Take a look at this slide and look at this carefully. Let's zoom in on this. Describe the concentration that you see in each of these. So maybe just press pause really quickly. All right, so here, if you look at the concentration, yeah, it looks kind of low, right? If you just had to describe that. And the water, therefore, is going to be drawn inside of the cell because this is what is called a hypotonic solution. You can think of these solute here, the salt or sugar, like a sponge, so the water will go in there. Now let's look at this next concentration. Look at this one carefully. Kind of looks equal, right? And you can see the movement of water. As much that goes out, the same amount goes in. Again, water's just going to naturally move, but it's going to move in equal amounts. So the cell doesn't expand. Over here, it's getting full of water. It gets really big. So this is actually called isotonic. Equal, just a fancy word for equal. Look at the last one here, C. Look at that concentration, really high, hyper. Notice what happens to the cell, what happens to the water. The water rushes out. Again, think of these solutes like a sponge. They will draw the water out. Also, if you think about it, there's more water inside this cell, so it naturally wants to flow out, right? So a high concentration of water, if you like, inside of the cell. Okay, we're going to look at this in greater detail. There's a bit of a lab that we're going to take a look at. So let's go to that YouTube video. It's in your link. I'll show it to you here. It's right here. It's going to take you about 5 minutes. So pause this video and then watch this YouTube video on egg osmosis. Again, the link for this video is in our description, plus it's in the Moodle course. Okay, so you've just finished watching the video. It took about 5 minutes there. What you might have noticed is that they were comparing basically how do they test whether a cell, whether water can move in and out of a cell. Well, first of all, it was not to drink it, right? And they did osmosis with an egg and ultimately dissolved the eggshell and of course ended up with that semi-permeable membrane. Notice what happened to A here. They took the mass of that one before it went in and it was 94 grams. But remember, how many grams was it when it came out? It expanded and it became 97 grams. Okay, so in that hypotonic solution, water rushed in and look at that egg, it got bigger. What they called B here, when they measured its mass before it went in, it was 103 grams. Then after, well, it shrank. I mean, look at it. Look how it's gotten smaller. It went down to 59 grams. So it got smaller. So the water got drawn out. So ultimately our conclusion was hypotonic. Water went in through osmosis. Hypertonic. So this one is hypo or low concentration. That's what hypo means. It means low. Hyper means high. So, you know, this is a corn syrup solution. Water came out. Again, movement of water, osmosis. So these 3 questions are going to be in your lab inside of Moodle. So let's take a look at these 3 questions that you need to answer. So, what was the mass of the egg and the one in the salt solution? Well, it was a corn syrup, so it'll be a corn syrup. And then the one just in the tap water, or distilled water. So again, the mass of that egg in the salt solution was 103 grams. The final mass was down to 59. And then the mass of the one in the regular water or the tap water started at 94 grams and went up to 97. And yes, you're going to have to be answering basically these 3 questions. What happens in a salt solution? What happens in the distilled water or the tap water? Either one will work. And then you're going to want to be able to draw what happens to a cell in a hypotonic, isotonic, and hypertonic solution. So that'll be the lab that you'll be handing in. But yeah, let's continue with this, with the notes here. We're going to take a look next at Yeah, particle motion. Yeah, why does this happen? Let's take a look at that. Sort of little delay there. Particle motion and the concentration gradient. Why does this even happen? How do particles even move? Ultimately, there's two reasons. First of all, particles are always in motion. Even if something is really, really cold, a surface or a metal is really, really cold, particles are always in motion. They could be going slow if it's cold, but they're still always in motion. And especially when you put something in water, it's going to move around fairly easily. Secondly, this is just a law of nature. They follow what's called the concentration gradient. And we all know this. Whether you burn some toast, whether you open up a perfume bottle, you have an area of high concentration, particles are eventually going to spread out, and eventually the whole house will smell like perfume, or the whole house will smell like burnt toast. So that's ultimately why this happens. Why do particles move? And we either get a diffusion of water, or we get a diffusion of oxygen into a cell, or a diffusion of carbon dioxide out of a cell. We've seen the concentration gradient. Let's take a peek here at this next slide. So yeah, some examples of passive transport. Again, no energy required. Simple diffusion. A couple examples going on there is going to be O2 going into your cells, CO2 gas going out. Osmosis, this happens in both plant and animal cells, so it's water, either in or out. And facilitated diffusion, yeah. Let's look at facilitated diffusion. This also requires no energy, but yeah, let's take a look at facilitated diffusion. Let's skip that slide. So it's the movement of particles across the cell membrane with the aid of transport proteins. Those proteins, this would just be a model of what they're looking at. But ultimately an example is what's going on here, exercise. So essentially their cells are needing glucose. Specifically, it's the mitochondria that needs glucose. Glucose is actually too big to just go across the cell membrane. It needs some kind of a channel to go in. And in fact, to open the door for glucose, insulin. That's the job of insulin. Insulin actually opens the door for glucose. So an example of facilitated diffusion. And again, you're going to want to write down the information that's on the screen here, not just what I'm writing, but all of this information. Facilitated diffusion example. Glucose gets into cell via a protein channel. Opened by insulin. That's insulin. It's a hormone. That's its job. That's pretty cool. And of course, that glucose, it's going to the mitochondria. So the mitochondria can burn it with oxygen to recharge ATP, which these people are definitely using up their ATP. So let's see here. We've seen an example. What do I want to do next? Active transport. Yeah, let's take a look at active transport. We've seen the types of diffusion that don't require any energy, just simple diffusion, osmosis, and facilitated diffusion. Next is active transport. So active transport does require energy, and it's pumps inside of your cell membrane, and they have an ability to basically pump materials in and out. This is also right here. This is endocytosis right here. So the whole membrane can flex and engulf materials, and this is exocytosis right here. we'll look at those two as well. These all require energy, but right here, what we've got, you've got your cell membrane, and this is literally happening right now. Not only the neurons in your brain, but also the neurons throughout your body. They have to basically set themselves up with a potential to fire off. So by pumping sodium out, it creates quite a potential. And when your neuron gets stimulated, it fires. Suddenly you see the color red. Suddenly someone pokes you with a pin and you hurt. But your neuron will fire. And the reason why it fires off is there's been a potential created by pumping sodium out. Anyway, main point, this requires energy. Because if we're going to get this much high concentration, this is against the concentration gradient. Look at that. That sodium just wants to rush in. And in fact, when the neuron is stimulated, it does just that. It rushes right through a channel, actually by facilitated diffusion. It goes back in, and then it gets pumped back out, so your neuron is ready again. Cool, right? All right. So lastly, finally, we do want to talk about endo and exocytosis, both also requiring energy. Let's take a look at these quickly. So endocytosis, it's when a cell takes in substances too large for passive or active transport. This still does require energy, of course. And so we've got some particles outside the cell being engulfed. You can actually see that happening right here. This is a macrophage, and basically it's destroying some used up red blood cells. This is actually happening in a spleen, essentially what's going on here. There's an amoeba eating something, some kind of food particle, or actually could be a small bacteria that it's trying to go after there. So again, this is when something is too big and it's got to get inside of the cell, and literally the cell membrane is so flexible, it'll rotate around it. So that's endocytosis. Let's finally look at exocytosis. The opposite, when we're sending stuff out. So this could be a protein that the ribosome made, because remember what ribosomes do, they make proteins, so in the cell. And it could even be the protein insulin that needs to get into the blood to then help sugar get into all of our cells. So that's an example of exocytosis. Okay, let's do a quick summary of what we've gone over today. So we've been looking at processes that don't require any energy just because of simple particle motion. You get a high concentration, things are going to want to come into the cell membrane either through diffusion or facilitated diffusion. We've also talked about osmosis. So one of the best examples is osmosis in plant cells, and you can have lots of water getting into the central vacuole, or that can dry out, and perhaps this plant has been stuck in salt water, or perhaps it's just in a really dry place. Again, those particles are just going to naturally move from high concentration to low and eventually balance out. And lastly, we saw exo and endocytosis. So endocytosis going in, exo going out. All right, so you can now complete that osmosis lab, and the next task after that is going to be doing the cell membrane assignment. We've got a review, and then you got a quiz, and then you got your exam. Okay, have a good day, Science 10. Talk to you again.